Hello, everyone, and welcome. I'm the host of today's online symposium, Jessica, from GenScript ProBio Marketing Department. On behalf of GenScript ProBio, I'd like to thank you all for attending today's symposium. GenScript ProBio is a subsidiary of GenScript Biotech Corporation, proactively providing end-to-end CDMO service, from the drug discovery to commercialization with proactive strategies, professional solutions, and efficient processes in cell and gene therapy, vaccine, biologics discovery, and antibody protein drug to accelerate drug development for customers. This online symposium will include 4 parts. In part 1, we will share a 10-minute overview introduction of ProBox platform, and for the extra 3 parts, from upstream and downstream process development to analytical development, each part will take 30-minute presentation and 10-minute Q&A session. You can submit questions at any point during the symposium using the chat or Q&A panel situated in the menu on the right. Now, I'm delighted to introduce today's first speaker, Dr. Ying Li, Director of Purification Process Development Department of GenScript ProBio. Dr. Li leads the CMC purification process development and provides technical leadership in new technology platform development. She joined GenScript ProBio in 2022, bringing over 10 years of biotechnology experience spanning multidisciplinary areas. Prior to joining GenScript ProBio, Dr. Li was an R&D manager in Merck KGaA from 2017 to 2022. Lead the development of viral vector manufacturing platform and biocontinuum platform. Dr. Li obtained her BS and PhD in chemical engineering from National University of Singapore and the Pennsylvania State University respectively. She is a frequent speaker at ACS and AICHE and has authored 8 publications in top-tier bioprocessing journals. For the first part, Dr. Li will share the overview of ProBox platform. Now, I'm delighted to hand over to Dr. Li. Dr. Li, please.
I hope you can all see my screen right now.
Not yet. Wait a minute. Okay, it's okay now.
All right. So let's get started. Hello, everyone. My name is Ying Li. It's my great pleasure to present today's topic, address CMC challenges with ProBio toolbox platform. Let's start with an overview of current global biologics market. In 2021, the global biologics market is projected to be worth over $330 billion, with a CAGR of 9.1% expected over the next decade, due to the broad application of biological drugs in a variety of indications. From the regional perspective, as you can see, North America still holds the largest market share in the global biologics market. And on the other hand, Asian Pacific is expected to be the fastest growing region in the global biologics market. In terms of the product segment, currently monoclonal antibodies segment still held the largest market share. However, there is a strong interest and trend towards the design of more advanced therapeutics to achieve improved efficacy through novel mechanism of action, including bispecific antibodies, antibody drug conjugates, recombinant proteins, cell and gene therapy, etc. ProBio, which is the CDMO segment of GenScript, has built a strong track record providing CMC services for all of these different biologics. This slide provides an overview of types of projects our platform has completed on record. As you can see, about 50% of our projects is related to mAbs, while around 25% is bi or tri-specific antibodies, with the rest of 25% related to fusion proteins and recombinant proteins. As we all know, these types of complex biological molecules pose significant challenges in the process development, from the upstream production to downstream purification and formulation. We have spent a lot of efforts in the past years to summarize lessons learned from the accomplished projects and developed our ProBox platform. Let's first take a closer look at the types of molecules collected in our ProBox platform. Starting from bispecific antibody, it could be broadly categorized into IgG-like or Fc-related bispecific antibody and non-IgG-like or Fab-related bispecific antibody. Under Fc-related bispecific antibodies, a typical structure includes symmetric bispecific antibodies, such as IgG fused with single-chain Fv or single-domain antibody. On the other hand, asymmetric bispecific antibodies, which include the classic structures, which could be assembled both in vitro and in vivo, and other different asymmetric structures. And here are some examples included in our non-IgG-like examples, such as Fab fragment or a single-chain Fv fragment fused with multiple single-chain Fv in the conjugation structure. On the protein side, we are proud of completing IND filing and GMP productions for a number of complex recombinant proteins. Here we categorize them into two different groups. The Ig fusion protein, which could be further divided into Fab fusion protein or Fc fusion protein. And in addition, quite a number of recombinant proteins have been studied in the past, including spike proteins, microglobulin, enzymes, or growth factors, etc. I would like to highlight the average titer and yield, which has been calculated for all the different molecules in the past. So for bispecific antibodies, the average titer achieved is up to 3.2 grams per liter under our platform, with average yield of 52%. And for fusion proteins, the average titer is 3.6 grams per liter, with average recovery of 44%. There are many unique manufacturing challenges posed for these complex molecules. For bispecific antibodies, for example, the generation of byproducts such as homodimer, aggregates, and fragments, those are difficult to be purified byproducts from the cell culture. And in addition, the low expression level and development of accurate analytical methods are also challenges faced for bispecific antibody development. On the recombinant protein side, in general, the low protein stability and complex PTM, the design of the capture steps, especially for proteins without any tag, removal of HTP, and of course, improving expression level and development of analytical methods such as bioassays are challenges faced in most of the recombinant protein projects. To address this problem, significant time and effort needs to be spent to identify effective solutions during process development, and sometimes the solutions may not be adequate. So here is an introduction of our ProBox process development toolbox. As I mentioned before, this is based on a lessons learned from over 100 ProBio CMC projects and integration of 200 challenges and solutions commonly faced in process development. And here is an overview of the ProBox. Can you still see my slide?
Thanks for Dr. Lee's introduction. Now, please let me introduce the second speaker, Mr. Rubin Zhang, Director of Nanjing Antibody PD and MSAT Department of GenScript ProBio. Mr. Zhang received his master's degree in biotechnology from Northeastern University in 2014. In the same year, he worked at Sanofi Genzyme as a process engineer for upstream process development of antibodies and recombinant proteins. From 2014 to 2020, he worked in the process development department of Merck Sharp and Dohme as a researcher and senior researcher, responsible for the upstream process development, process characterization, and validation, as well as leading the upstream process development of Merck Sharp and Dohme's continuous production and process online analysis upstream development. During this period, he participated in the publication of 7 academic articles. From mid-2020 to 2022, he worked in Hangzhou Yi'an as Deputy Director and Director, responsible for Upstream Process Development Department, in charge of CDMO, Upstream Process Development, and also promoted and enhanced the feedback process and continuous perfusion process platform. In this talk, Mr. Zhang will discuss ProBox solutions for upstream process development. Now, I'm delighted to hand over to Mr. Zhang. Mr. Zhang, please.
All right, thank you for Jessica's introduction, and thanks very much to Dr. Lee to have a brief introduction about the ProBio's technology platform. Now I'm going to talk about the cell line development and upstream process development toolbox for complex biologics. Here is an overview about the platform toolbox for the cell line and upstream. Our toolbox includes vector design, which including codon optimization, signal peptides, which signal peptide to choose, and the vector system for the bispecific antibodies. And also we have topics about transfection. We also have topics about clone selection, including how to do the single clone confirmation and what is the selection strategy for high titer and good quality clones. We also have a very good strategy to do the clone stability study, which can balancing the time for development and also the clone stability. For upstream, a lot of times the topics includes titer increase, quality attributes optimization, intensified fed batch, and perfusion cell culture. For upstream, for scale up, a lot of times we may have a lactate spike or low cell growth and a low QP problem when we're scaling up the process. So these toolbox is comprehensive review of all these vectors. Today, I'm going to choose some of the topics from the toolbox. So this is today's topics. First section is about the titer improvement. As we all know, a good cell line is always the foundation for success and high titer upstream. So I'm going to talk about our CHO genes and apt vector system from the GenScript ProBio. After that, I'm going to talk about the dual vector system for bispecific antibodies, which can greatly increase the productivity and also the purity. The third one I'm going to talk about is upstream parameter optimization, followed by intensified fed batch process and perfusion process. For N-glycan modification, which is another big part for upstream, I will talk about how to increase the galactosylation, how to decrease the fuel oscillation, and how to increase the cellulation for long half-life product. On the third big part, I'm going to talk about enzyme activity improvement, and last but not least, I'm going to talk about the scale-up issue. So for each topic, I will have solutions and case studies. Again, for the overview for cell line and the cell culture process development, when a new CMC project come in, it always start with the cell line construction. At this stage, we want to produce high productivity clones using CHO cell genes and a tighter up vector system. The next step is always clone selection. We're using 24 well plate for clone and Ember 15 for clone selection. In the same time, we also have a comprehensive strategy to guarantee the clone stability while balancing the development time. For media screening, GenScript ProBio have a top-tier media brands that are available for quick tests and get it ready for use in the process. For bioreactor process development, we would focus on the process optimization of temperature, pH, culture duration in 3 liters. In the same time, if our clients would need a higher titer, we would develop the intensified fed batch for the specific project. And if we have unstable product, we would recommend to use a perfusion process to develop the unstable products. When scaling up, ProBio have a proper scale-up strategy to guarantee the comparable performance between the pilot scale and the 3 liter. And also ProBio have its own good GMP production system that can meet the quality requirement for both Europe, US, and mainland China. Now I'm going to talk about the CHO-K1 genes and up vector system. The CHO-K1 genes is GenScript ProBio's in-house host cell line. We have a commercial use license from ECACC, and the cell was adapted to serum-free suspension culture in ProBio. And coupled with the host cell line, we have our tighter up vector system. We insert the transcription active region. This vector can insert into the transcription active region of the chromosome. In the same time, it can speed up the stable pool stage, which can shrink the development timeline for cell line development. It can significantly improve the titer of difficult to express molecules, and we utilize this vector for map by specific antibody and protein productions. On the left bottom, you can see the difference between our classic vectors and the vectors using the up vector system. Different from the classic ones, we actually have a quite high producing clones from the beginning. And in the middle of the graph, you can tell that we not only have 1 or 2 high producing clones, but each transfection using the vectors new vector system can harvest multiple high producing clones from the transfection. This actually provides a solid foundation for high producing upstream process. The up vector system was start in service starting from the end of 2022. So we're still accumulating data, but now I want to review some of the data from our CHO-K1 genes host cell line coupled with our classic vector system. On the left bottom, we can see the average titer for different protein modalities. For monoclonal antibody, on average, we have 5.4 gram per liter. And for bispecific antibody, like what Dr. Lee mentioned, the average titer is 3.2 gram per liter. And for recombinant protein, the average titer is 3.6 gram per liter. And in terms of the distribution on the right figure, for monoclonal antibody, 10% of the project will fall in between 2 to 4 grams per liter, and 40% of the product will fall in the category between 4 to 5 grams per liter, and 40% of the project will fall in the category between 5 to 8 grams per liter. And luckily, we have a more than 10% about the project can reach more than 8 grams per liter, even without any cell culture optimization. The next question after the cell line development is naturally goes to the stability of the cell line. ProBio guarantees more than 60 PDL stability for the project from the TROK1 genes and the TROK1 ADCC platform, and we guarantee there's no sequence variation in genetic level. How much is PDL 60? Would PDL 60 enough for the large scale? The figure in the middle actually answers this question. We set the primary cell banking as a PDL 0, and for master cell bank, the PDL is 12, and for working cell bank, the PDL is 24. After we saw a while and start to scale up, if we scale up to 200 liter, the PDL will be 40, and at the end stage of 2,000 liter, the PDL will be 43, and at the end stage of 8,000 liter, the PDL is 46. You can see there is still a long way to go between the PDL number we guaranteed. In the same time, our host cell line have excellent run-to-run comparability. On the bottom figure, I'm showing one example that we have a continuous 3 runs in line, and each run all the runs have a similar viability, similar titer, and a similar product quality. Let's switch gear, talk about a little bit about bispecific vector system. The reason I mention it is because for bispecific manufacturing, we always have some monospecific impurities. Sometimes we have a really low product yields and high aggregation concentrations, and some of the undesirable fragments. To solve that problem, ProBio actually developed a 2-vector system to improve the productivity and the purity for the bispecific products. The difference, like what I showed on the left bottom, instead of using 2-vector, we're actually using 2-vector system, and for 2 chains, we separately copied them into different vector. Then we adjust the vector ratio before the formal transfection. The first transfection is more like a try to find out the best ratio between different vectors. Then we do the formal transfections according to the vector ratio we learned from the last round. By using this strategy, we can greatly decrease the homodimer and increase the heterodimer productivity and purity. This is a real case from our projects. On the top, I'm showing the vector system using the single vectors. From the SDS page, you can tell our target product is chain A plus chain B. However, quite some of our product from this transfection are chain 2, chain B, or only chain B alone. And this data further verified by the SEC HPLC and NRC SDS, and the cell poop fed batch production titer was only 0.5 gram per liter. On the bottom is the 2-vector system transfection after the ratio optimization. You can see from the SDS page, the majority of the product are the correct form, and this number is also verified by the SEC and NRC SDS. If we analyze the case on the top, you can tell there's a large amount of chain B compared with the chain A expression. Because of that, during the transfection in the transfection of the dual vector system, we would greatly increase the vector number for the vector concentration for chain A, but decrease the vector concentration for chain B. So eventually we can balancing the number of the chains of the two chains produced. By doing that, the cell pool production titer was increased from 0.5 gram per liter to 5.4 gram per liter. After the cell line, I'm going to talk about the upstream process development. During upstream, how could we increase the titer? The way including do media screening and feeding strategy development. Also, sometimes additives would help for the cell culture performance. In terms of the process parameters, we can always optimize temperature, pH, and even DO level. If the common measures wouldn't help for the titer, we can also develop perfusion process or intensified fed batch process if we want to greatly increase the titer. Here is a good example about temperature optimization enhance the productivity. Internally, this data was organized from the 11 projects. There are literature saying behind the product temperature decreasing, there's a literature explaining the principle behind it. So after the temperature shift from 37 to lower temperature, 33 degree or 30 degree, the lactate metabolite was greatly optimized. And also the cell cycle was arrested at G1 phase, which stopped the cell from growing, but changed the cell from growing to production. In the same time, the metabolic was also changed from lactate generation manner to a lactate consumption way. The lactate would go into the TCA cycle and also phosphate pathway and also glycolysis cycle. By doing that, especially for low expression molecules, the titer was greatly increased. On the bottom right, you can see for MAPS, after the temperature optimization, the titer can increase to more than 30%. And for non-MAP product, a lot of times it can greatly increase to 172%. This is another great example of the downshift can increase the productivity in the same by enhancing the productivity and also increase the duration of the cell culture. This case is from SCFV expression. This molecule itself have really low expression in our platform process. However, we start process development with 3 different temperature. On the left bottom, you can see at 30 degrees, the cell only reached 15 million cells at peak cell density. And at 33 degree, it reached around 18 million cells, and at 37, it reached 20 million cells. However, as the temperature goes up, the viability actually drops faster. So at the 30 degrees, the viability can maintain around above 80% on day 15. If we look at the titer data on the right, for titer, the 30 degree titer actually increased more than around 6-fold compared with the culture which kept at the 37 degree. And in terms of the QP was also greatly increased comparing with the default 37 degree process. Since at the low temperature, we also have a really good viability. So to further increase the titer, this process was the process duration was extended from 15-day process to a 17 days process. On the top middle, you can see the product quality attributes. Starting from the day 15 and day 17, the product quality attributes are quite similar. However, starting from the day 19, the product quality starts to getting worse. So the process team decided to stop at day 17 as a final process. This process was scaled up to 250 liter, and after the 3 runs was quite comparable with different scales. Finally, before the before the process optimization, the titer was only 0.3 gram per liter, and after the process optimization, we successfully increased the titer to 2.06 gram per liter. This is a good case for high feeding increased titer. So this is the IgG4 case. During the platform process, the viability was fine and the titer was fine. However, for our platform, as a platform process, we also test a different feeding strategy. For high feeding strategy, which have around 30% of the feeding volume compared with the basal media, it increased the average titer from 6 gram per liter to 8 gram per liter, even though there's no impact to the cell growth. As a result, by increasing the feed volume, we successfully increased the productivity from 6 to 8, and there was no influence on cell growth for IgG4. If we want a significantly increase for productivity, the common measures may not work, so we would use intensified fed batch to greatly increase the titer. Different from the conventional fed batch process, at N-1 stage, we will use perfusion. By doing this, we can greatly increase the inoculation cell density from 0.5 million cells to 10 or even more 20 million cells per ml from day 0. And this will shorten the production cycle. In the same time, it will greatly increase the titer. The figure in the middle is actually highlights the different 14 projects that use the intensified fed batch. On average, the production titer was increased more than twofold, and in some cases, it's increased more than threefold or even reached the fivefold. Here I'm using a FC fusion protein case. This is a FC fusion protein, and it has a high concentration formulation, which require high titer. So we start process development in both fed batch mode and the intensified fed batch mode. For fed batch mode, the inoculation cell density was 0.5 million cells per ml, and for intensified fed batch, the inoculation cell density was 10 million cells. From the figure in the middle, you can tell the titer was increased from on average 6 gram per liter to more than 10 gram per liter. And after the process optimization, the highest titer reached 15 gram per liter. On the right bottom, you can tell there's no quality attributes difference between the fed batch process and the intensified fed batch process. Another way to increase the productivity is using the steady state perfusion process. Here, I'm using a showcase of the IgG1. This culture duration was up to 84 days. The productivity was about 28 times compared with the traditional fed batch. In traditional fed batch, the productivity was 4 gram per liter. And at the end of the 84 days, the accumulated titer for perfusion process was 110 gram per liter. And the quality attributes was stable during the steady state phase for this perfusion process. Here I'm showing a real case for a FC fusion SCFV case. For this project, 2 SCFV was added to the bottom of the protein. And this product suffers from the low expression level, and also in fed batch process, after the one-step purification, there was still very high HCP remaining in the process. So the process was developed and to a perfusion process up to 27 days, and the productivity at the end of the day was increased by 10 times. For fed batch process, the titer was 0.78 gram per liter, and for perfusion process, the cumulative titer was 7.7 gram per liter. On the bottom left is showing the cell growth profile, and on the middle of the bottom is showing the productive activity profile, and on the right bottom is actually showing the greatly decreased host cell protein number. For intensified fed batch, after protein A, there are still around 3,000 HCP. However, for the perfusion process, the number was greatly reduced to 400, and for drug substance, it was further reduced to 20 ppm. And glycan is also one big part for upstream for the product quality attributes modification. There are common requirements like fuel constellation modification, galactose modification, and the salicylic acid modification. Here I'm showing a case, an IgG4 case to increase the galactosylation modification. By adding the galactosylation modification, we can improve the binding activity. That's why the clients would need to increase the galactosylation. There was several components was tested, including some commercial available reagents and also literature available components. After the try, galactosylation was increased from 3% to 28% with a mixture of trace elements, nucleoside, and also the substrate. There was no significant influence on titer CX and also MEN5. For the principle behind it is very easy to understand. So galactosylation is also an enzyme activity. In order to optimize this enzyme activity, we actually find three factors that actually can help. The first is the substrate of the concentration of the substrate. Therefore, we add more galactose into this media. In the same time, there are always cofactors for the enzyme, including some of the trace element and also some nucleotide. That's why we added them in. And eventually, the galactosylation level was increased to around 30%. Well, it has no impact to other N-glycan profile, especially the MEN5 was kept low. The other part I want to talk about the fuel constellation reduction. Clients may have needs to increase ADCC activity. There are actually two ways to, in order to increase ADCC activity, so fuel constellation level need to be bring down. There are two ways to do that. The first one is the best one is always to modify the cell line directly. At ProBio, we actually developed one cell line that can knock out the FU8 gene to produce afucosylation antibodies. The FUT8 enzyme is actually responsible to adding the fucosylation to the protein. And after we knocked out that enzyme, fucosylation cannot be added to the protein. And the cell line we actually have it is CHOKI1 ADCC plus cell line is a stable cell line for CMC service with a comparable productivity and also clone stability. On the right, you can see the fuel constellation level using the ADCC enhanced cell line compared with other commercial available additives to decrease the fuel constellation. For the best additives from the commercial available reagent, it can decrease the fuel constellation to 35.1%. However, for ADCC enhanced cell line, there is no fuel constellation can be detected at all. However, for the clients already have their cell line, there's no way that we can modify that host cell line in short period of time. The other strategy is to use the additives to decrease the fuel constellation. This is a good example for a IgG1 project. So additives were added to the cell culture medium, and the fuel constellation content decreased from 98% to 35%. There was no influence on titer and other quality attributes, including MEN5. On the bottom, you can see the MEN5 content was constant between different conditions, and the titer was comparable for the additives B and the control. And by doing that, the fuel constellation, the ADCC activity would actually increased more than tenfold by decreasing the fuel constellation. In an interleukin projects, we actually add additives to raise the glycosylation. When there's no glycosylation in the interleukin, the protein becomes unstable. If you look at the right up figure, on the one doesn't have Na there, that means that specific molecule don't have any N-glycan there. And after 40 degrees for 2 weeks, that product actually quickly degraded to 3 different main peaks. And for the ones with N-glycan on the top, they are stable for 2 weeks at 40 degrees. And the glycosylated interleukin showed much higher stability. And also, it was reported that cellulation prolonged the half-life of the protein, like the interleukin. So for this project, we're looking forward to increase the glycosylation in the same time, increase the cellulation as well. Again, different components, additives were adding to the basal media in order to increase the glycosylation. And we have trace elements, nucleoside, and also the substrate were adding to the medium. The cellulation was successfully increased from 42% to 90%, and glycosylation was increased from 80% to 98%. And most of the interleukin were glycolized, and the non-glycolized protein was not even detectable. After optimization, if you look at the upper right figure, you can see all of those has N-glycan on them, and in the same time, which is more important, is there the 3SG, the NAL, NAC 3SG peak becomes much larger. That means the cellulation content will also greatly increased, which will help the half-life. Let's switch gear, talk about the enzyme activity. So we have one project that is a lysosome enzyme case. So for this enzyme, it needs to be modified between release to the environment. Otherwise, this enzyme will not be active. So during our upstream process development, we find out as the duration goes up, and also as the titer goes up, the specific activity of this enzyme actually goes low. So it has a reverse trend of cell culture duration and the specific activity of the self-taste. Later on, so we developed a perfusion process in order to increase the specific activity of this product. On the upper right figure, fed batch process and perfusion process was compared. For perfusion process, the cell density was reached 60 million cells. And for the specific activity, the activity was increased from 30 to 300, which is a tenfold increase just by switching from fed batch process to a perfusion process. In order to further increase the activity, we also find out the cofactor of the enzyme. The two trace elements were also added to the cell culture, and the concentration of these two trace elements were determined using a DOE study. And in the bottom middle, you can see the activity was increased in the DOE studies. Some of the conditions actually reached 900. And so at the end of this process, the enzyme activity was increased about 30 times after the cell culture optimization by switching the fed batch to a perfusion process and also adding the cofactor trace elements into the media. The last but not least, I'm going to talk about a little bit about the scale up. For a lot of our process, if the cell line is from non-platform cell line from the clients, then scale up strategy need to be optimized when running the cells. Because when scale up, depending on the metabolism activity of the cell, some of the cell may more sensitive to the environment, and we may see the lactate spike during the process. On the left bottom is a real case happening in our pilot plant. The lactate spiked in the middle of the process. And in the following round, we actually mitigate the issue by adjusting the agitation, DO, and the macro sparge. And the lactate metabolism came back to historical trend in following batches. For scale up, there are several factors we also wanted to consider. One of the most important thing might be the DO level, and also nowadays, more and more cells have a high cell density, which reaches 30 millions or even 40 millions. So the P188 concentration is also a good factor to optimize for our process. That's all I have for today. Now I'm available for questions. Thank you a lot.
Okay, now please let me invite Dr. Li again for the third part sharing. In this talk, Dr. Li will discuss ProBox solutions for downstream process development. Let's welcome Dr. Li.
Thank you, Jessica.
You're welcome.
I hope you can see my screen right now.
Yeah, it's okay.
All right. So hello, everybody again. So under this session, I'm going to talk about downstream process development toolbox for complex biologics. So first, let's start with an overview of common challenges faced for purification process. So starting from depth filtration, there are a number of problems related to antibody reduction or protein absorption, and followed by capture step. And in general, the capture of antibody without Fc fraction or protein without tag could be problematic to design a capture step. And in particular, some unstable proteins may be difficult to be captured or eluted, especially at low pH elution conditions. And similarly for VIN, for proteins unstable at low pH or unstable in SMD, we need to identify strategies to develop this VIN step. And in addition, for pollution chromatography steps, typically including a number of ion exchange, CEX, AEX, and hydrophobic interaction chromatography, and mixed mode chromatography, the removal of some difficult product-related impurities, such as aggregates or fragments, could be a big challenge. And for some antibodies, and in particular some proteins, the removal of difficult HCP is a big challenge. And well, at the same time, to improve the overall product recovery. For nanofiltration, in general, the low loading capacity and low stability protein are common concerns for this particular unit operation. And finally, for UFDF, the high concentration formulation pose significant challenge to the design of this unit. And here is an overview of the downstream toolbox. So here we covered quite a variety of different problems as I just talked in the last slide. So we find in general this is a quite effective way to improve the development capability and shorten the development timeline. So here is an overview of today's topics. I'm going to select a number of problems that most people have expressed concern with purification and formulation for biologics, which include reduction, capture, how to deal with aggregates, homodimer, remove of HCP, and finally high concentration formulation development. So let's start with the first challenge, the reduction of antibodies or proteins. So here is a case study we have accomplished in the past. So this is a fusion protein with isoelectric points of 7.8. And upon development, we identified this molecule is quite unstable upon storage in the room temperature when we first harvest the supernatant. And here you can see from this graph, upon harvest, the main peak, the CE purity is up to 98%. Well, when the supernatant was stored at room temperature for 7 hours, there was a significant generation of antibody fragments, including the 2 heavy chain, 1 light chain, or some other impurities. And the main peak purity dropped to 56.7%. Therefore, this molecule has high reduction tendency. And increasingly, we have seen a lot of cases like this, even for monoclonal antibodies. So in general, the antibody disulfide bond reduction is essentially a redox reaction that involves redox enzyme. So this particular reaction needs to be categorized by CO-endoxin reductase, and also requires CRX, and also requires the NADPH to provide electrons to kick off the reaction. Therefore, there are a couple of methods based on this mechanism to reduce the reaction and reduce the reduction of the molecule, which includes the use of microions or chelating agents, which will inhibit the redox reaction by reduce the reactivity of the enzyme. And oxygen or some other oxidants could consume the NADPH through a competitive reaction pathway, therefore to shut down the reaction as well. And in addition, minimize the cell lysis, therefore reduce the generation of the enzyme, lower the temperature, and pH can also slow down the reduction reaction. And our team have built a reduction evaluation platform to systematically evaluate and mitigate the risk of reduction. So under this case, we applied a few different strategies, which include continuous air sputtering for overnight, adding trace amount of copper ions, and or adding low concentration of histamine. And you can see from the results in comparison to the control case, there was a significant reduction of the generation of the 2 heavy chain, 1 light chain impurities by using all of these different strategies. And finally, we selected increasing dissolved oxygen method, because this is something very easy to be implemented in a large-scale manufacturing process. Next challenge is about capture. Like I mentioned, in general, the capture of antibody fragments without Fc fragment, and also the capture of recombinant proteins without any tags would be quite challenging to be done. And for the first case is related to a Fab fragment with a PI of 8.7. And here, this is integral Fab molecule. And for this particular case, our team screened different binding domains using different affinity chromatography resins. In particular, for Fab, there are 3 different domains we can potentially utilize, which include the VH domain, the VL domain, and CH1 domain. And there were a number of resins available in the market that we could potentially select from. And upon screening, we identified that for most of the Fab molecules, utilizing VL domain is the most effective way to capture the Fab fragment, and in particular with much higher capacity compared with using other binding resins. And in comparison, we screened a number of different capital L or protein L resins and identified an optimal one with a significantly improved overall capacity, which again reduced the overall cost for the manufacturer. And through this one-step capture step, we successfully achieved SEC purity about 99.5% with over 96% of C purity. So lessons learned is always select the best affinity resin for the right binding domain, and also screen different resins to get the highest capacity. And here is another case, which is a nanobody CHH fragment fused with an enzyme. So there were a number of challenges associated with this molecule. So first, again, this molecule does not contain any Fc fragment, so this is difficult to be captured. And in addition, because of the enzyme fraction, this molecule is quite unstable, so it tends to precipitate when pH drops to below 7.0. And this could be seen in this chromatography graph when we elute the bond products with using the normal acetate buffer at pH 3.5. We found the majority of the products were aggregates of fragments, and when adjusted to pH 7, the pH adjustment did not resolve aggregates, and the precipitation still exists. So for this particular case, first we did a comprehensive screening study to identify the optimal VHH binding resin, which was selected as our platform IC resin for all the nanobody related products. And after that, we also did an optimization for the elution process. In particular, we changed the elution buffer from acetate to glycine HCl at the same pH. So in general, it will still be able to elute the target protein while the eluent was at much higher pH, close to neutral at pH 7.2. So here it avoided the pH adjustment across the PI and also storing the protein at low pH, therefore results in a highly purified product with SEC purity above 97%. Next, let's talk about the capture for no-tag recombinant proteins. So for this particular case, we're dealing with a giant protein molecule, which is a tetrameric protein, and also at the same time at pretty low PI at 5.9, and this is not uncommon for a lot of recombinant proteins at acidic PI. So the challenges include, again, first, this you cannot find any affinity resins, in particular for this type of recombinant proteins. And again, this protein is highly unstable. So especially we did a screening study to identify a design space for keeping the molecule to be stable, and identify that under low conductivity and low pH, the protein tends to be form precipitation, and therefore cannot be further processed without product loss. And in addition, without effective affinity chromatography step, the HCP level is quite high. And therefore, to solve this problem, we first identify the right capture chromatography to capture this recombinant protein molecule. We end up with a CEX. This is in general our recommendation because utilizing AEX for the first capture step, sometimes you will end up with capturing the majority of pigments, which will result in blocking of your chromatography column. For utilizing the CEX, here we need to balance the stability and the recovery to choose the best loading conditions. Remember that we just mentioned there is a very narrow design space for this type of recombinant protein. And after that, we need to investigate the different pH and conductivity sensitivity between HCP and target protein, and we choose pH dilution in this particular case, which was effective in removing the target protein, diluting the target protein while removing the majority of HCP as well. In addition, a wash 2 step at pH 6.2 was added to further wash out the HCP, and arginine is added to the elution buffer to eliminate the abnormal pH rise and further reduce HCP. As you can see, upon all this optimization with the wash 2 step and arginine addition on the elution step, the overall recovery is up to 92% with 88% of ICC purity and significantly reduced HCP level. So the next challenge is about aggregates. This is one of the biggest challenge in a lot of bispecific antibody development. So in this particular case, I include the case for the IgG fused with single chain FV. Because of the exposure of the hydrophobic region of the single chain FV, this is not uncommon to observe a significant percentage of high molecular weight present in the intermediate, supernatant and intermediate products. So for this particular case, as you can see, after the affinity step, there was still 8% of the high molecular weight, which was quite challenging to be removed. And upon optimizing AC, including the wash and elution step, or the CEX step, cannot remove aggregates effectively. So under this situation, we generally recommend using a mixed mode chromatography. So for this particular case, a mixed mode AEX in flow through mode was utilized for high molecular weight removal. And we adjust the optimal loading pH and conductivity to achieve the best separation of monomer and the high molecular weights. With one step chromatography, will achieve a final ICC purity above 98%. Again, in our experience, utilizing multiple interactions, including hydrophobic interactions, electronic interactions, or hydrogen bonding, will significantly improve the separation for some difficult to be removed impurities, such as aggregates. So second case is also a bispecific antibody. So this case is an asymmetric bispecific antibody. Again, this molecule was designed in a knob-into-hole structure to prevent mismatch. And unfortunately, there was significant high molecular weight present in the AC alloys, around 20%. And we tried using different CEX resins, and also at the same time, some mixed mode CEX as well. And none of them achieve a pretty good separation of the aggregates and the MNP. At the end of the day, we utilize the CHT, which is a ceramic hydroxyapatite chromatography, which is a crippling manual of calcium and phosphate, which contains a casein site with high metal affinity and phosphate site, which is a classic CEX mechanism. In general, this chromatography is quite effective in removing a lot of high molecular weight. And there are a lot of tips to optimize this particular chromatography step, including the maintaining operation pH at a relatively high pH to extend the lifetime of the resin, and make sure to include phosphate in our samples and buffers at a base concentration to improve the resolution of the separation, and combine the elution with both phosphate buffer and sodium chloride buffer to get the best result. Again, some other tips include, again, of course, adding wash 2 to remove free peak fragment to improve the separation of the monomer and high molecular weight. So here, as the results shows that upon using one step of CHT, the high molecular weight drops from over 16% to less than 1% in this one single chromatography step, which is quite effective. And next challenge, again, which is highly relevant to bispecific antibody, is the removal of homodimer. Ruben just talked about reducing the expression of homodimer from the optimization using the viral vectors, especially adjusting the different ratios of viral vectors. From downstream perspective, in general, some of the mismatch of homodimers could be removed by some of the chromatography steps. So here is one example of asymmetric bispecific antibody. Even though a knob-into-hole structure was utilized, there was still significant amount of homodimer, the whole homodimer expressed in the cell culture, and it is up to 40%. And this is a quite big challenge for downstream purification, especially considering the similar chemical and physical structure normally observed for the two chains. And the capture and intermediate purification are not effective in homodimer removal. So in this case, we selected mixed mode chromatography MMC for homodimer removal by both the charge and hydrophobicity difference. So here, upon again utilizing MMC, there was almost all the 40% of the homodimer was successfully removed with final purity about 99% in the final alloy, as you can see in the graph. One of the key for the success for this particular case is the relatively big difference in terms of the PI between the two chains. Upon analysis from ICIEF, we could identify the delta pH between chain 1 and chain 2 is close to 1, which is considered to be pretty significant. Therefore, it is quite effective utilizing MMC or cation exchange mechanism to remove the two different mismatch species. And for HTP removal, this has been a big challenge for a lot of complex biologics, in particular for recombinant proteins. Because many recombinant proteins have relatively low PI, which is close to the PI range of HTP, making the removal of HTP difficult. And at the same time, there is no existing affinity resin for capture of many recombinant proteins, therefore leaving high level of HTP post-capture step. Oftentimes, this level is about 10,000 ppm. And on top of that, as I mentioned before, the proteins are generally unstable under certain pH and cell conditions, therefore further limiting the operation space for most chromatography steps. So in general, the solution again could start from upstream. So in our platform, we always select the optimal host cell system. In the ProBio system, in general, for the majority of the molecules, the starting HTP level upon capture or affinity step is below 2000 ppm. So that gave us a pretty good starting point. And again, the controlled cell culture process, including duration, VCV, and viability of the cell culture, could also help resolving some of the starting HTP level for downstream purification. So from downstream perspective, we have developed strategies to optimize the wash and elution conditions for a number of chromatography steps to effectively remove HCP species. And in general, we recommend using the hydrophobic interaction chromatography utilizing hydrophobicity difference to remove those HCPs, such as using HYC or MMC chromatography. And finally, most of the time, we will use intermediate DF step, which utilize the filter, optimize the filter and loading conditions to significantly absorb a lot of HCP species. Here is one example of Fc fusion protein. This is actually a factor 8 blood factor, which was again expressed with high HCP level and quite unstable molecule. So here is an overall path for removing the HCP level from over 60,000 ppm down to less than 20 ppm through 4-step chromatography, or 3-step chromatography step process. So starting from the AC, the starting HCP level was quite high. And after that, the utilization of HYC was quite effective because part of the HCP with stronger binding ability will be removed from this particular step. And we compared 2 different routes going through the AEX directly or adding an intermediate DF step filtration step, and identified that the AEX effectivity could also be significantly improved by adding intermediate DF to reduce the starting HCP level, which brings down the final HCP level to less than 20 ppm. And finally, this high concentration formulation is again one of the biggest challenge in the entire downstream purification downstream process, and including both purification step and formulation step. There were a number of challenges associated with high concentration DS process. The pressure drop was typically much more significant compared with lower formulation conditions. And generally, the yield of the process at UFDF step could be impacted. And sometimes a pH excipient drift could be observed due to the high concentration of protein expressed in the final formulation buffer. And of course, the formation of protein aggregation, especially under the conditions when the proteins are not stable. So there were a number of underlying root cause for this problem, including the high viscosity of the solution, the dominant or volume exclusion effect, and the self-association with the protein molecules. And all underlying that was highlighting the intermolecular interaction of the protein molecules, especially under high concentration. So here, a number of cases we successfully developed strategies to address the challenges in high concentration formulation. The first case is a bispecific antibody with PI of 8.5. So for this case, the DS concentration is required over 120 milligrams per milliliter. And we observe a pH drift at higher protein concentration as seen in this graph. So the target pH, final formulation pH is around 6.5. But when we add the proteins, exchange proteins into target buffer, the pH was drift significantly above the target value. And therefore, we developed a strategic platform to compensate the effect of the pH drift and identified a different buffer for the buffer exchange step in order to compensate the pH drift effect. And also the increased viscosity results in the pressure challenges. And in general, you need to take a closer look at the optimization of the TFF step before and after the change of the buffer and at different protein concentrations. So in this particular case, the optimal operation flux, feed flux was also switched when the protein concentration was increased and the diet filtration buffer was switched from initial buffer. And also for this particular case with the fusion protein and again with low PI acidic protein, the DI's concentration requirement was over 140 milligrams per milliliter. And in particular, the overall concentration step, the concentration of the protein reached above 200 milligrams per milliliter. The increased viscosity, of course, will result in a lot of pressure challenges. So in this case, our team selected the optimal viscosity reducer in order to reduce the viscosity of the protein formulation. Here, as you can see from the table, adding sodium chloride was quite effective in reducing the viscosity of the formulation. As you can see, at high sodium chloride concentration, even for very high protein concentration at about 150 milligrams per milliliter, the viscosity level was below, was at 11 CP, which was much easier to be processed in the USDF step. Therefore, in general, it is recommended to select and optimize the viscosity reducer and adjusting the concentration of the excipients to reduce the concentration, the viscosity of over-concentration step. And of course, adjusting the inlet flow pressure and appropriately select the membrane cassette with in particular for the over-concentration step. So in summary, hopefully I have demonstrated today there were a number of unique challenges posed for complex biologics, such as bispecific antibodies and recombinant proteins that require significant time and efforts to search for effective solutions. And here at ProBio, we have developed a comprehensive platform to address typical challenges faced in both purification and formulation unit operations. And with that, I would be more than happy to take any questions you may have. Thank you.
Okay, thank you for Dr. Li's excellent presentation. There are really many great case studies. Now, please let me introduce the next speaker, Dr. Mengping Liu, Senior Scientist for Quality Study Department of GenScript ProBio. Dr. Liu leads the CMC Analytical Development Service for protein and antibody and provides technical leadership in new instrumental analytical development. She obtained her MS in pharmaceutical analysis from Sun Yat-sen University and PhD in biomedical science from the University of Hong Kong. She published 5 first author papers in well-known SCI journals and is dedicated to the quality study of biologics. In this talk, Dr. Liu will discuss ProBox solutions for analytical development. Now, I'm delighted to hand over to Dr. Liu. Dr. Liu, please.
Thank you for Jessica's introduction.
You're welcome. You can share your screen.
Yeah, sure. Can you see my screen?
Yeah, it's okay.
Hello, everyone. My name is Mengping Liu. I'm the senior scientist of quality study from GenScript ProBio. Yeah, it's my pleasure to have the opportunity to introduce some analytical solutions from ProBox for biologics CMC challenges. In today's presentation, I'm going to first give you a brief introductions of the ProBio analytical platform, and followed by the solutions, analytical solutions from the ProBox developed for analytical and process development problems. Finally, I will end up this presentation with a short summary. First, please allow me to give you a brief introduction of our analytical platform. Our platform could provide multiple analytical services for our clients, including the early drug developability assessment, quality control in CMC, drug characterization, and extra. We have rich experience of developing over 200 analytical methods and have analyzed over 30 complex biologics, for example, biospecific antibodies and fusion proteins. In addition, our platform is equipped with advanced instruments, for example, the mass spectrometers from the Q extractive, UPLC, HPLC from Indolent Waters, Thermo Fisher. Yeah, just guarantee we could provide reliable and high throughput analytical service. This table presents some analytical methods we developed in our platform. Basically, for each quality attributes, we have at least 2 methods just to ensure our results could be confirmed with different techniques. And we have alternatives in different scenarios. For example, for title measurement, we have developed ELISA, octet, and protein A HPLC methods. As we know, the quality study for protein therapeutics is always challenging because these molecules have complex structures and by activities. Here, we list some typical challenges we analyze in biospecific antibodies and recombinant proteins. For biospecific antibodies, they are usually expressed with low titer level. And so we need to develop very sensitive methods for titer measurement. In addition, the design of the bioactive assay is very difficult because these molecules really have varied MOA and multiple targets. In addition, besides that, biospecific antibodies are usually expressed with a number of mismatched byproducts. For example, the homodimers for asymmetrical bispecific antibodies. These homodimers usually have very similar physicochemical properties to the main target, to the target molecules, making it difficult to analyze and identify these homodimers. And that further brings problems for purity analysis for these molecules. And then for recombinant proteins, and the same problems we found as well. In addition, these molecules usually have a complex disulfide shuffling or post-translational modification. And that also leads to the challenges of analyzing the molecular purity. Because of that, and then we developed a ProBox platform. This is our view of the analytical solutions in the ProBox. platform. And today I'm going to focus introducing some analytical solutions, particularly developed for analytical development and CMC troubleshooting. First, please allow me to introduce some solutions from the ProBox for analytical development problems. The first type of problems is the low response signal in the analytical methods. And in this case, we found very low response signal in the when developing a non-reduced CSDS method for antibody. Well, this antibody the stability of this antibody was evaluated in a low pH stability study. Yeah, we found it showed a pretty low signal only after 2 and 4 hours incubation under the low pH condition. That's unexpectedly. Yes, and we analyzing its electrophoregram and found that aside from the low signal of the analyte, also we can see the low signal of the marker, indicating the method itself has problem. We further investigated the buffer system of this method and found that the buffer contains high salt. As we know, the high salt actually can lead to low injection efficiency for this method, particularly when we used electrokinetics injection method. And so that would lead to lower loading sample to the system. Besides that, the salt ions in the buffer can interfere with the negative charge of the analyte in the running in the system, and that might lead to the inhibition of the response signal as well. And then because of that, and performed the buffer exchange of the sample to a low salt trace buffer. And now we can see after the buffer exchange, the signal of the analyte was back to normal as well as the markers, as well as markers. So that this solution get this problem solved. Second type problems is the low resolution in some analytical methods. Well, the case relates to our IgG1 antibody. Here, we need to develop a high HPLC method for this antibody. We first tried our platform method by using the column 1, but we found that this column was incapable of reading the oxidized products from the IgG1. And then we switched to using another column 2. Yeah, for this column, it filled with ligand B that shows higher hydrophobicity than ligand A in the column 1 as the solid phase material. Besides that, the particle size was reduced to 2.5 micrometer. Yeah, with this parameter optimization, we found now the resolution of this method it's improved significantly. We can see the good separation of oxidized and non-oxidized products for the IgG1 antibody. So for this kind of these problems, we should consider to use appropriate ligands and smaller particle size. Yeah, for a resolution improvement. And the third type of problems we would like to share with you is related to the poor quantification accuracy for some analytical methods. Here, the case in this case is about the development of competitive binding ELISA AC. We found a very high heel slope in this AC, indicating that this AC is not very accurate for measurement. Well, here when we developed this ELISA assay, we tried different binding formats. First, we tested the format A. In format 8, format A, the anti-spike is the analyte, and ACE2 is the receptor, is the target, and the spike is the competitor. We first immobilized the ACE2, the target molecules on the plate first, and then added anti-spike and the competitor spike into the system, allowing them to interact with each other. Here shows the binding curve of format A. By analyzing its heel slope ratio, we found that the ratio of value is most of them is out of the system stability test range, indicating this assay, the results of this assay is not reliable, and yeah, it's not very sensitive and to some errors in the middle. We guess the reason is that when we immobilize the ACE2, the target on the plate, some active binding domains may not be able to be exposed and to the analyte anti-spike. And yeah, due to some, for example, the steric hindrance effects. And then we switch to format B. In this format, we immobilized the spike, discompleted molecules on the plate first, and then added anti-spike and its target ACE2 receptors into the system. In this way, the analyte and the targets can freely bind to each other. Wow, and here this is the results. Now the heel slope ratio of this binding curve is right in the system stability test range. So indicating the method is more reliable. Okay, and then let's go to the next part. Here, I'd like to introduce some analytical solutions we developed for process development problems. The first type problems we usually met is the molecular cleavage identification. Molecular cleavage is very common, particularly for fusion proteins. And in this case, we are going to talk about the FC fused protein. For these proteins, and is prone to be cleaved during the cell line development stage. And here are the possible fragments of these molecules. And for our analytical department, we should develop a very sensitive method to monitor the molecular cleavage, particularly in very early process development stage. Here, we choose to develop the Western blot assay because this method is very sensitive and specific, and that can sensitively detect as low as like 0.1 nanogram of a protein, and could use specific antibodies and to identify the possible fragments for this fusion protein. Here shows some details of this method development. Well, we choose to use these two specific antibodies targeting FC and VHH segments of this molecule. And here shows how we're gonna interpret the Western blot assay data. For the full length of protein and the fragments with both VHH and the FC segments, they can all be detected by the FC and VHH antibodies. But for the individual fragments, just with VHH or FC segments, and then they can only be recognized by their specific antibody. And then we use this Western blot assay and to analyze the proteins expressed in the cell pore 165. The bands in the red square are the proteins from the pore 165. Aside from the full length of protein, we can also observe the another two bands. Analyzing its molecular weight, we guess that probably these two types of fragments. So this data basically suggests that the proteins from the pool 165 should have significant cleavage. So for this type of problems, we suggested using, for example, we suggested using a sensitive method to enable early monitoring of the molecular cleavage. For example, the Western blot. For the second type of problem is is to identify the post-translational modifications. Yeah. During the process development. This case raised to a IgG1 antibody. By analyzing its high chromatogram data, we can see that the main peak was splitted into splitted in the middle. That indicating these products has some variance in caused by different hydrophobicity. And we guess that might caused by the post modification of this molecule. For the antibodies, and there are three major types of PTM, including the C-terminal lysine clipping, N-terminal phytoglutamic acid cyclization, and glycosylation. For this molecule, the glycosylation level is pretty low, and yeah, usually cannot be detected by the high chromatography. So we guess probably caused by these two type of PTM. Well, and so and then we proposed analytical strategy for this problem. Okay, and first is to use the specific enzyme, CPB and a QPCT, to achieve complete lysine clipping and pyroglutamic acid cyclization to see whether this enzymatic digestion can eliminate the the heterogeneity caused by these two types of PTM. And aside from that, I'm gonna further separate these two peaks and analyze them with LC mass spectrometry. Okay, and this is the results. After the enzymatic digestion, we can see the two peaks become only one single peak. Yeah, so indicating the heterogeneity was eliminated, and also kind of confirming the heterogeneity in previous product probably caused by the lysine clipping and glutamic acid cyclization. To further confirm that, we separated the peak 1 and peak 2 and purified them for further mass analysis. This is the data from the mass analysis. So basically, you can see that, well, peak 1 and peak 2, they indeed have the PTM modification as we hypothesized previously, and they are mainly formed in the heavy chain, the antibody. But the percentage of these two peaks are kind of different. For peak 2, it have almost 100% of PTM, but for peak 1 is kind of lower, and low percentage of these two types of PTM actually leads to a lower hydrophobicity of the sample as well, and that can explain why peak 1 is eluted earlier in the high chromatography. Okay, so And for this kind of problems, yeah, sometimes we can use specific enzyme and to have some, initial ideas of the type of PTM in the molecule. Yeah, with this information, we can have better guidance and for further study. The third type of problems we met in the process development is the investigation of potency loss for some molecules. This case relates to IgG4 antibody. During the formulation process development study, we found the potency of the molecules significantly lose over 50% only after 2 weeks of storage at 40 Celsius. And usually the loss of the potency of the sample can be caused by the sample degradation or some undesirable PTM. Okay, so and then we proposed this analytical strategy for this kind of issue. We first analyzed the purity of the sample to see whether some degradation cleavage happened. Later, further analyzed the PTM or fragmentation in the sample with the acid mass. Here's the results. This is purity results. We can see that the non-reduced CESD data suggests that in front of the heavy chain molecule, there's possible fragments from the heavy chain. That's also observed in the SDS page gel data. With this information, and they will perform the RP-HPLC mass analysis for the sample. And here, the data suggests that the sample, the heavy chain of the sample, indeed was cleaved. And this fragment observed in the CSDS, probably from the G50 to K443 of the heavy chain fragment of the heavy chain of this antibody. And in addition, we observed oxidation, this kind of PTM in the heavy chain fragments. And the level is over 70%. So basically, we can see probably the heavy chain degradation and the oxidation lead to the decreased potency for IgG4 in the formulation development process. For this type of problems, we suggest them to investigate the molecular structure, from whether some fragmentation or undesirable PTM happened in the molecule. For the last type of problems we are going to talk about today is about the misassemble identification. Here we give I give a case related to extreme medical bispecific antibody. This bispecific antibody is formed by two chains, chain 1 and chain 2. By analyzing its ICIF profile, we can see that there's a acetic peak 1 in front of the main peak. The level reached to approximately 2%. We should identify this peak 1 to see whether it's a home dimer and which type of home dimer it is. So the strategy is to purify this peak first and then send that for a mass spectrometry analysis. Wow, but when we performed the peak purification, found it's quite challenging by using just a HIG or CX chromatography. And we guess the reason is because the physical chemical properties of this antibodies is quite similar to the target molecule. And so next, we tried another strategy, purification strategies by using a mixed mode chromatography. here, they combined the affinity capabilities from the CX and high resins. Now get a more purified peak for mass analysis. And this is a mass data. And found that the acidic peak 1 is indeed a home dimer, and that's formed by two trend tools. So for the misassemble identification, particularly for the mismatched homodimer from the bispecific antibodies, we can consider using new purification chromatography, for example, the mixed mode chromatography for to achieve for better purification efficiency. Well, in the end, it's a summary of today's topic. So basically, from previous CMC projects, we found it's quite difficult to analyze the molecules, for example, the bispecific antibodies and recombinant proteins, because the complexity in their structures and by activities. But now, GenScript ProBio developed a ProBox, and that has collected a set of effective and up-to-date analytical tools designed for analytical development and CMC troubleshooting, and that we believe can efficiently accelerating the CMC development for our clients in the future. Thank you, and welcome any questions.
Thank you for Dr. Liu's excellent presentation. Let me join others in thanking Dr. Li, Mr. Zhang, and Dr. Liu for their time today. Thank you. Also, thank you for your attending today's online symposium. Look forward to meeting you again soon. I'll send the recording of this symposium to all registrations through email next week. You can check the recording next time. Thank you.